Now it's my pleasure and honor to introduce our first keynote speaker, Professor Tony Charman from King's College, London. Professor Charman is chair in clinical child psychology at the Institute of Psychiatry, Psychology, and Neuroscience at King's College, London. His main research interest is in the investigation of social cognitive development in children with autism and the clinical application of this work via screening, diagnostic, epidemiological, and intervention studies. He's a chartered clinical psychologist and works in a specialist service for children with autism and complex neurodevelopmental conditions. He has published more than 350 peer-reviewed papers and over 30 book chapters. He has served on a number of expert panels for the Medical Research Council in the UK, the National Institutes of Health in the US, and the WHO. He has also worked with various organizations to advocate for services and positive policy development for individuals with autism spectrum disorder and their families. Today, his talk will be on getting answers from babies about autism. Please join me in welcoming Professor Charman.
Thank you, Patrick. Can you just confirm that you can see my slides again now?
Yes.
Thank you, Patrick, and the committee for inviting me. It's an absolute pleasure. Obviously, we all hope this would have been an in-person meeting, but you know, I'm sure we'll get the chance to meet again in sort of future. So I'm going to be talking about some of the work that I've been doing with my colleagues here in the UK. I am focusing quite a lot on studies from the group that I work with. Many other groups are also looking at infant siblings, as we often refer to the design with the family history of autism and increasingly other sort of conditions. But unashamedly today, I'm presenting work that mostly originates from our group. I'll just put up my declarations of sort of interest because they're there, research grants, some consultancy and some sort of royalties. I will also just mention I'm in my office. It's a real background behind me, and actually weirdly the Wi-Fi in my office is worse than in my house. But if I fall out, it'll only be temporarily because it just often blips out and I have to come back in. But thank you for joining me this afternoon, and I'm looking forward to speaking to you. So here's an overview of what I'm going to be talking about. Just briefly talking about what we've known for quite a long time, for 20 or 30 years now, about early accounts of how autism might manifest or develop in young children, and then talk about studies that have been being conducted around the world now for about 20 years, which I've called here the new science of autism in infancy. These have changed, and I'll talk about the changes in terms of their focus. A lot of the focus early on was on identifying initial differences in infancy, in infants who would later go on to have autism. I think now there's a more complicated set of sort of aims or goals or strategies to sort of research, and I hope to outline some of them that at least our group, as well as many other groups, have been sort of taking. I'm going to be talking a little bit about how we and other groups now are looking at what we call shared and distinct pathways in the infant developmental sort of period to not just autism, but later commonly co-occurring conditions such as ADHD and anxiety. I'm going to talk a little bit about some of the models that we and others have sort of developed that we think are going to be important going forwards to have a more mechanistic approach to understanding early developmental differences. And briefly at the end, I'm going to touch on some intervention studies that we've been sort of running to see if it's possible to change the early course of development in infants with a family history of autism and indeed later on ADHD. This is an old slide now from Geraldine Dawson, but it sort of shows you where we were 15 or so sort of years ago in terms of the sorts of things that people were looking at in the experimental literature in young children with autism. Much of the research at the time was being done in in toddlers, 2 and 3 year olds, with an early diagnosis of autism. And people were looking at a number of behavioral and experimental sort of paradigms, but also we're interested in some of the underlying sort of neural circuitry and things such as sort of neurotransmitters and other sorts of things. Lots of the focus around that time was on early social development, with the understandable sort of expectation that the core defining characteristics of autism are social communication and social interaction difficulties, along with rigid patterns of repetitive behavior and thinking and sensory differences. But the social communication impairments have always been the core sort of diagnostic feature that defines the condition. And we knew that that could be recognized and seen, and in fact is part of the diagnostic criteria for a very young child. And that's where the focus was. So these are some reviews that came out some 10 plus sort of years ago now, which was sort of summarizing some of the work that had been done in the previous 10 years, because the first studies of infant siblings who have a brother or sister with a diagnosis of autism were started something like 20 years ago. I mean, the first study was actually started by Simon Baron-Cohen and Nira Yamira and Marion Sigmund back in the mid-1990s, but the first large programs started some 20 years ago. So these are summaries, but they're sort of telling us what we knew sort of 10 years ago, and I'm really taking us from that point in time to what we've learned over the past 10 years. So broadly, we know the population prevalence of autism in children, at least around the globe from many studies. This is a UK and a US sort of study, but there are many studies. It's probably somewhere between 1% and 2%. That's not the topic of this talk, but that's a broadly accepted prevalence figure for how common autism is, at least in childhood. What we know from large population studies, particularly in the Scandinavian, Northern European countries, is that the sibling recurrence rate, the likelihood of 2 children in the family both having a diagnosis, is something like 10%. So that's 10 times higher than the population prevalence rate. It happens to be that in the volunteer families, who have been engaged in research, primarily in North America, the US and Canada, but also here in the UK, and we have colleagues we work with in other European countries as well. So, the recurrence rate in the families who are being studied, perhaps because they're volunteers, is perhaps as high as 20%. So the prospective longitudinal design, where we recruit infants very early on in development, indeed we often recruit parents before their new babies are born. The way that most of us do this is that we recruit parents because they have a young child with a diagnosis of autism and they're having another baby. And it's an efficient enough design. If the sibling recurrence is something like, in population studies, 10%, or in volunteer studies, perhaps higher, perhaps as high as 20%, families are willing to take part, they are motivated to take part, they stick with us. And it means that if you're studying a cohort of children, of 100 children, who are infants, who have got an older brother or sister with autism, then it may be that 10 or 20% of those infants will go on at some point later on to develop autism. More recently, and I'll come to this later on, we and other groups are also looking at infants with a family history of ADHD and genetic conditions. These are slides from Sally Osnos's group from UC Davis in California, from the Mind Institute. And this is an old slide. There is a more updated one with a much larger sample, but this, I think, is a very sort of nice sort of description, where here the infants in their lab are seen at 6-monthly intervals, and they're just showing you here the trajectory on social orienting behaviors. This is gaze to faces, social smiles, directed vocalizations, and a rating of social engagement. And here, when they see the infants at 6 and 12 months, the lines between those who go on to have an autism diagnosis at 3 and those who are typically developing are crossing. It's only after the 12-month period that the differences emerge. And some of these differences do seem to be actual declines in social orientation, in social engagement, in social smiling, and a lack of the increase in vocalization seen in the typical infants. So that's a very good summary of what we know about how early the social orienting impairments seem to emerge. They don't seem to clearly be there before the age of 12 months, but after that they become apparent. And obviously it's around that 2 to 3 year period that many, but not all infants can be diagnosed. This is a different slide from Warren Jones and Army Klin sort of group here, where they've got a very intensive design where the infants are seen in the first 6 months on a monthly basis, and then they're seen at 3 monthly intervals after that through to the age of 18 months, and then 24 months, and then later on at 3. Again, this is an old sort of slide, but I think it's interesting and somewhat different pattern here, because here they're looking at the infants who later go on to have an autism diagnosis versus typically developing infants. And they're seeing here, this is fixation time to eyes on the scale here. And they see here that there are differences that emerge where the fixation to eyes is lower in the infants who go on to have autism, again, perhaps from as early here as 9 months of age, so somewhat earlier than on the Ozonoff slide that I showed you before. What's also interesting, and I'll come back to this later on, is that if anything, it looks like the infants who later go on to have autism are looking at the eyes more at the very earliest stages. This is at 2, 3, 4, 5 months of age. So it may be there's a kind of reversal sort of going on where there may be atypical looking patterns in terms of fixation to the eyes in these video sort of stimuli, but that may be, there's a suggestion that may be higher at the very earliest developmental time points, but then becomes lower, which is more consistent with the pattern and the clinical presentation that helps diagnose the children when they're 2 or 3. So this is a summary of our sort of study. It's confusingly sort of called a number of different things. For about sort of 10, 12 years, we were calling it the British Autism Study of Infant Siblings, BASIS. More recently, for the past 7 or 8 years, we've been studying both infants with a family history of autism and ADHD. So that's called STARS, Studying Autism and ADHD sort of risks. So we call the sort of cohort basis STARS. Just in brief, although most of the infants are recruited on the basis of the fact they have an older brother or sister with a diagnosis of autism or indeed ADHD more recently, we also do recruit infants whose parents have a diagnosis of autism or ADHD. Other groups are using that similar design as well. And then we see them, and this is a collaboration with Mark Johnson, who used to run the Baby Lab at the Centre for Brain and Cognitive Development at Birkbeck. He's now the head of department in Cambridge and is still obviously heavily involved. Mark and I set this program up more than 15 years ago. Emily Jones runs that project at Birkbeck in the Baby Lab there. And they see the infants at 5, 10, and 14 months of age, and my group see them here at King's College London at 2 and at 3, and now I'll come to this, we're seeing the children up until the age of 10 to 12 at the moment. So these are the same babies followed through over a period of getting towards now 10 years. These are the groups. We've changed our language because the community seemed to, or at least some in the community don't like the word risk. So we talk now confusingly about elevated and typical likelihood. Many of my slides are from previous talks, so they haven't been updated, but I'll switch between the two. And then we look at outcomes in relation to both diagnosis of autism and potentially ADHD, but also, as you see, a lot of other traits of individual differences, atypical development, anxiety, emotional dysregulation, and many other psychopathology sort of outcomes. So this is a sort of summary of the work we've been doing for the past sort of 15 years. So, I mean, the story is, we've seen something like getting towards now 450 infants through this program, and that's taken us 15 years. So, these are long and complex studies to run, with a whole army of researchers and students and postdocs, and as you can imagine, it's a huge sort of task because of the multiple time points at which the infants are seen. I'm leaving some things out to make it more sort of simple, but these are the sort of cohorts, and then we see them through those infancy phases. We see them in mid-childhood. We also get data, both phenotypic data on parents and brothers and sisters, and we get genetic data, which I'll mention briefly, on different family members. I'm going to mention these two intervention trials we've embedded within our last two cohorts. One is targeting social communication development in autism, and one is targeting cognitive development in infants with a family history of ADHD. This is just sort of saying what we do, and it's now a very complex set of sort of methods that we sort of use. We gather information about family environmental factors. We gather parent-child interaction data from live interactions, led by my colleagues at Birkbeck. We use a number of different imaging sort of techniques. I'm not going to be presenting MRI data, but with colleagues here, with David Edwards' group here in London, we do both neonatal and 4 to 6 month MRI, but I'm not presenting that data today. We also have near-infrared spectroscopy. We have EEG. We're using accelerometries and electrophysiology and skin conductance and motion detectors. We have eye tracking experiments, behavioral experiments. We have lots of developmental and clinical sort of measures, and we are collecting cheek swabs and saliva at different sort of ages. So this is one of our earlier sort of sort of findings that led by when Maya de Elzebah, who's now in McGill, was running the sort of studies sort of for us. And this is sort of showing an ERP experiment looking at dynamic gaze in terms of a video with a model's face gazing towards and gazing away from the infants. And this is data from 7 months of age, and these are the outcome groups. The low risk or typical likelihood controls, those at high risk or elevated likelihood who don't have ASD, those who have autism spectrum disorder, and those who had autism probably even as early as 2 years of age. And here what we find at 7 months of age is there's an attenuated response in the neural response, and this is the amplitude in the neural response to dynamic gaze shifts in the infants who later on at the age of 3, this is 7 months of age, go on to meet our research criteria for a diagnosis of autism. Here, from a different experiment conducted at the same time, here's the proportion of time looking to the eyes. It doesn't differ between the groups. So we're able to sort of demonstrate here that, as in some other studies, at 7 months of age, the looking behavior, the social orientation doesn't look different, but it may be that the neural response, the processing of that social stimuli, the dynamic gaze stimuli, is attenuated in those who later go on to have sort of autism. We'll come back to some of those sort of findings. Here's another study sort of led by Mayada on the same sort of initial sort of cohort. It's a classic gap task with baseline trials, and then, sorry, this is out of line, but overlap trials where the peripheral stimulus appears either in the absence or in the presence of the fixation stimulus. So here's the disengagement cost, the difference between the saccade to the peripheral stimulus in the baseline versus the overlap trials in this particular sort of metric. And here are the autism groups in terms of their outcome at 3 years of age that broadly don't differ at 7 months. But here we see a quite surprising, remarkable finding in many ways that actually the other groups, as we'd expect, the disengagement cost as the infants are maturing goes down. So the speed of saccade between the two different stimuli at 14 months is lower in these groups. But in those who go on to have a later diagnosis of autism, it actually increases. So development is going in the opposite direction to what you would expect. This is a different sort of paradigm. There are videos of an actress, and she's doing it. We call this peekaboo as a sort of experiment. So the actress is doing sort of peekaboo, or just and singing into the video with the infant in front of them with an eye tracker, or they're moving their eyes only, or they're moving their mouth only with a nursery rhyme type sort of song. Here, here's the data in terms of the looking sort of behavior. That is the eye mouth sort of index here at 7 months and 14 months. These are the 4 outcome groups in terms of the infant siblings who go on to have autism or other atypical outcomes, or who are typically developing, or the low risk typical likelihood controls, and the looking pattern across the different paradigms, particularly where the peekaboo condition or the eyes moving only condition, the looking pattern is not different across the groups, regardless of whether you go on to have an autism diagnosis or outcome. So these early findings, at least for us from our own experiments, were beginning to make us think that even at an early age, we were not finding basic social orienting differences, certainly before the first year of life. There have been some, and I've shown you the Army, Klin, Warren, Jones sort of finding, but more findings from our group and others seem not to show at least pervasive social orienting impairments in the infants who go on to later have autism before the age of 12 months. Here's a different study led by sort of Sam Waz, and this is looking at fixation durations. It's actually fixation durations to a facial stimuli and a pop-out task. And here it's showing that those who go on to have autism have shorter fixation durations. We find a similar finding to non-social stimuli in some of our experiments as well. This is in relation to an outcome diagnosis, and here is in relation to a standard diagnostic measure conducted at the age of 3. So we're finding also differences in non-social attentional findings here in terms of fixation duration, and previously I showed you on the gap task. We also, and I'm not, I don't do EEG experiments, but my colleagues sort of do. So I won't walk you through the sort of detail here, but here's an EEG experiment looking at repetitive non-vocal tones. And we're finding here differences in reactivity, indeed hyperreactivity to repeated tones in infants who go on to have later ASD, go on to have autism at 3 years, as early as 8 months of age. This is the pop-out task, and here again at 7 months and 14 months, we're looking here, this is the proportion of time. There are many metrics you can derive from this task, but this is the proportion of time looking towards the sort of faces. At the age of 7 months, the groups, depending on outcome, do not sort of differ, and spend roughly 40 to 50% of the time looking at the face out of the stimuli. It's the most interesting for young babies. By 14 months of age, in those from the high risk or elevated likelihood sort of families, those infants are spending more time than the low risk controls looking at that face. So the decline is much steeper here in the typical likelihood or low risk controls than it is regardless of autism diagnosis in those with a family history. So here there seems to be, at least within all the infants with a family history of autism, there seems to be a different pattern of looking where they're spending here again longer rather than shorter looking at social stimuli when they're presented with the choice in the array. Then we follow up the same infants to 3 years of age, and here's a face recognition sort of task where the infants are the toddlers are presented with, the toddlers at 3 are presented with the choices about which is the women, which is the lady that they've seen before, and this is the face recognition proportion of trials correct, and here's the same 14-month variable in terms of proportion of looking time. Those who are looking more to the face in the pop out at 14 months of age are performing worse at age 3 years on the face recognition task. So it seems to be the longer looking is telling us something about atypical processing of faces as early as 14 months. They're not looking less, they're looking more, but that may be an underlying sign of a processing difficulty, which seems to be manifested in a behavioral task that requires a response. It's a touchscreen task, I think, at the age of 3. We then see the same infants. This was followed through to the age of 7 here, led by one of my postdocs who's now in Brazil, but Lizzie Shepherd. So here, these are data from the 7-year experiments. We use similar face recognition tasks. We have a face non-face ERP sort of paradigm, and we find that both face recognition skills and neural responses to faces versus non-faces is related to both reaction time in a false choice face recognition task, but also related, this is a social responsiveness scale, so a sort of dimensional scale of autistic symptoms at the age of 7. Then we can look back and see what these infants were doing at 7 months of age. So these are infants who have been followed for nearly 7 years, 6.5 years. And we can see that some of the infant ERP responses are related marginally to autism symptom severity. There is some continuity between the infant face versus noise ERP signal and that for the lateralization of the M170 and the P1 latency at 7 years of age. So there's some continuity both between the neural responsiveness at a group level and how that relates to later skills such as face recognition and indeed possibly even autism symptoms. So what I haven't done in this is to sort of cover much work by other groups, and there is a lot, and I've just put some of the more recent sort of reviews sort of down here, and there are many groups. I mean, this is my sort of one slide sort of summary of something like 300 papers. It's not systematic, but when do we see differences in those who later go on to have autism? Well, social orienting, but most of the studies only find that after 12 months of age. Some studies have found early repetitive behaviors in play with objects as early as 9 months, one study even 6 months, but there are many fewer studies on that. There's some suggestion there may be early sensory motor abnormalities detected as early as 6 months, but again, there are relatively few studies. Language development appears to be lower, but probably not until 12 months of age. certainly has been some work by Chuck Nelson's group and working with Helen Tarker-Flusberg in the Boston sort of labs, where they're finding some differences in EEG and connectivity and power from as early as 3 or 6 months age. Sample sizes are much smaller. We have shown some differences in reduced neural responsiveness to social stimuli with Sarah Lloyd-Fox's group in using NEARS from as early as 6 to 7 months of age. And there's led by Joe Pivin and others, the large IBIS consortium in North America, the infant brain imaging study, who have followed something like 350 infants with sequential structural MRI scans, have found a number of atypical brain growth patterns between 6 and 12 months of age. So that's a summary of things that relate to later autism. So if I count that sort of as the first wave of studies, what have we learned? Well, I think the first thing, and there are several sort of good reviews, including one by Mark Johnson and Mayada Elzabah from a few years ago, I think in Current Biology, but I can't remember, that where lots of us first started, actually, you have to look back and say that lots of what we expected has not come to pass. So what many people, including myself, expected was you would find, if you were using behavioral, but particularly experimental techniques, brain imaging techniques, eye tracking, looking behavior, you would begin to find a very early emergence of atypical social responses, essentially reduced social orienting and reduced looking to things like eyes in the very early developmental period. That's broadly not been the case, both because sometimes you find, both from the Army, Klin, Warren, Jones finding in the first few months of life, and from several of our studies that I've showed you, we actually find greater orientation to social stimuli in the first year of life, which may in itself be atypical. It's also true that there are very clearly other differences in terms of the distribution and focus of the attentional sort of system to both social, but also very clearly non-social stimuli, as well as other things such as habituation to auditory tones that appears to be different early on. So the sort of social first deficit accounts of autism are not supported in the simple way. One of the other things that's been a challenge for the field is that we and many other people often report single experiments in isolation. And you either do or you don't find sort of differences. People are only just beginning, and we've done this a few times, and I haven't shown you examples, I apologize, but combining different measures from different experiments to see if there may be a multiplicative effect in terms of how predictive they are, but also, as I'll show you in a moment later on, thinking about more complex mechanistic models of how we understand the perturbation that develops over time in these infants who may go on to have atypical outcomes. I mean, you can read papers where they report things such as sort of the sensitivity and specificity of some of these early assays to later autism, giving sort of positive predictive values that are very high. And if in fact one weekend you're wanting to do something that's work related but doesn't really feel like work, you can go to the US Patents Office and look at the number of things that are up there, including from very eminent researchers, where they have patents for particular indicators that may be early biomarkers of later sort of autism, and there are many. Of course, one thing that's also changed in 20 years is our view of genetics, which I don't really have time to talk about, and it's not my area either today, has changed quite a lot. There seem to be more and less heritable forms of autism because what we know about the fact that the majority of the genetic influence is many small genes, small genetic variants of small effect. And then there are other larger genes of effect and things such as copy number variations that for individuals, have a large effect, but may not run in families quite in the same way. So that story is still sort of unfolding. Um, um, have I just, yeah, sorry, I've got two sort of similar slides. One of the things that we've been trying to do over the past sort of 6 or sort of 7 years is to sort of be less interested in what predicts an autism outcome, because we know, I'm a clinician, as well as a developmental scientist. So we know clinically that co-occurring mental health difficulties are the norm in autism, not the exception. Some of our work shows in population studies from my group that 70% of children with a diagnosis of autism meet criteria for a co-occurring psychiatric condition. Autism does not occur neurodevelopmentally in isolation. It overlaps with lots of other things. And I've particularly mentioned sort of ADHD and anxiety because they're the most common co-occurring childhood conditions, but I could mention others. So in some ways, we've begun to move away from focusing just on what predicts later autism diagnosis to try and understand how developmental pathways lead to later characteristics that may mean that you meet criteria for a diagnosis of autism or ADHD or some other condition such as an anxiety disorder. But we're wanting to look at that all in the round. We're trying to use multiple methodologies, and I'll give you some sort of examples of that, where we're trying to combine things such as experimental measures such as EEG and eye tracking with genetic information, with phenotypic or clinical behavioral sort of outcomes, which we're just beginning to sort of grapple with now. A lot of the focus has been on what predicts autism through to the age of 3, and I haven't got much on that. But one of the things that's become clear for those of us who see the children now into mid-childhood, and in my lab, now we've after 15 months of no data collection due to the pandemic, we have been seeing families for the past 4 months, which is great, and we are seeing 12-year-olds who have been through the whole procedure. So focusing on a single time point, both sort of reifies the notion of sort of diagnosis being necessarily a sort of unitary and stable thing, but also misses the important developmental opportunity of thinking about these longitudinal studies as one way to really test mechanisms. And that's what I'm going to be talking a little bit about now. So this is a model that's really led by Mark Johnson, and Emily, our collaborators. Andrew Pickles, a methodologist, statistician who works with me, here at King's on this and many other parts of my, clinical research sort of program. And this came out at the beginning of the year, which is our sort of idea about how we can think about risk factors that could be genetic, like infants with tuberosclerosis or infants with neurofibromatosis 1, NF1, as we call it, or an autism or ADHD familial risk. And then we think about early sort of atypical development and some more sort of posterior early stage processing things, and then thinking about, we've focused here on these two in this model because these are ones that run through our program now. We focus both on social engagement and on what we call executive attention. So we have measures of these built into the longitudinal design, and then at 3 and through to mid-childhood, we measure a number of dimensional sort of traits. And we're looking here at the important modification of early risk, where early risk, whether it's genetic or familial, may mean there's differences or perturbations in early stage processing. And we can measure that using a whole number of different sort of experimental designs of the sort I've been sort of talking about. We're interested here in looking at the interaction within more sophisticated sort of structural equational type models, sort of, analyses, where we look at the modification and interaction of these over development. That's partly because if it turns out to be the case that executive attention and social engagement can can modify early risk that comes either from your family history or from being born with a particular genetic anomaly, then that may be important for thinking about targeting intervention. And I'll mention that as well. Here's a different model. Again, this was led by John Constantino, but he kindly involved Emily Jones and sort of myself, again, published this year again, which is somewhat different, but it's really here sort of emphasizing that there may be overlapping sort of risk pathways in terms of genetic contributions to things that we later think know, in fact, from family histories relate to later autism. Early on in development might be quite overlapping with other things such as ADHD and motor development. So how are we, how are we trying to sort of take those, how are we trying to take those sort of models and to sort of use them in practice? Well, here, as I've mentioned, there's a number of groups, us and other groups, who are looking at infants with a family history of ADHD, infants with tuberous sclerosis or neurofibromatosis, as well as infants with a family history of autism, and we're doing that in different studies. Here's another sort of take on sort of what we can see through to mid-childhood, again by Lizzie Shepherd, my ex-postdoc. And here we're looking at infant constructs. These are temperamental measures from the IBQ. And we're looking then at continuous phenotypic measures of anxiety, ADHD, and autism. And what we find is, that actually the infant predictors of later ADHD are really distinct from those that predict later autism, but there's quite a lot of overlap in terms of early fearfulness, which we see as an early sort of behavioral inhibition, a precursor really to anxiety. There's quite a lot of overlap between anxiety and sort of autism. So we're seeing here, there may be pathways that are more shared between later conditions such as anxiety traits and autism traits, and other ones that are maybe more distinct between early activity measures and later ADHD traits that don't overlap with later autism traits or anxiety. That's important because clinically we know that the rates of ADHD and anxiety are the most common co-occurring conditions we see in children with a diagnosis of autism. Here's some work by postdoc Anna Gui from Mark and Emily's lab that we're using the pop-out task again, and we have peak look duration to pop out at 14 months. Here we have familial liability of ASD and ADHD here in just under 300 families. We're looking at polygenic scores, polygenic risk scores, in the same sort of sample as well. And we're looking at clinical traits of both autism and ADHD at the in those who have been followed to mid-childhood, 6 to 8 years at this time point. And then what we see here is you just look at genetic liability for ASD and ADHD. And here, this is on the peak look duration in the pop out at 14 months. We see that longer peak duration is elevated in toddlers or in infants, 14 months, with an elevated genetic likelihood for ADHD, but not for autism. So here, the familiarity seems to tell us something. It's a very small effect size, but it's significant about later looking sort of behavior. And then we look at the looking behavior here at 14 months, and does that relate to that infant when they're in mid-childhood to their ASD traits or ADHD traits? It relates, yes, to ADHD traits and marginally to the autism traits at 7 years. So we see some sort of continuity in a rather complex way between genetic and familial liability and experimental variables of looking behavior and 14 months and mid-childhood phenotypic outcomes at 7 years of age. So that's a sort of summary of that. Here's just one slide from some paper that came out a few months ago led by Amy Goodwin. And this is just sort of, sorry, it's a bit hard to sort of read, but here we're showing our new cohort, the STARS cohort, which is essentially 161 infants with a family history of ADHD and autism, because many family members can have both. or different family members can have the different diagnoses, and those with a family history of autism only, and those a typical likelihood, the low risk, the ones with no family history. And this is a whole range of things. This is the parent IBQ. This is a consensus code from the experimenters. This is at 10 months of age of activity during the testing session. This is peak look duration during pop out. This is reaction time variability in the gap task. This is heart rate. This is head motion. We also have other EEG measures and other things. But anyway, the story at 10 months is that the infants who have a family history of ADHD don't look different on these measures, which surprised us as well, because we assayed about 14 things and none of them were different between the group who had a family history of ADHD and those who did not. We do find some continuity, both by parent report and by observer rating, of things such as activity, but not attention to later ADHD traits at 3. So we do find some meaningful developmental associations, but not the ones that we maybe expected. So again, fewer groups have been studying infants with a family history of ADHD using this design. So we're still in the early days of understanding what our findings, might be telling us. Here's one of our former PhD students, Mutlahan Ersoy. And this is just looking at anxiety. It's a rather complex, but also beautiful structural equational modeling that I won't try and sort of walk you through. But it's sort of talking here about effortful control in the temperamental measures, behavioral inhibition, fearfulness, as I've talked about before, and how that relates to later anxiety, which it does. And as we've shown before through to mid-childhood, it also relates to later ASD features. But we also finding interactions in these cross-lag structural equational models where it may be that executive control plays some kind of mediating role between early anxiety traits and later anxiety traits. That's the kind of thing that we're talking about in the amend sort of modifier sort of model that we've proposed. Here's another sort of recent paper from a few months ago, I think September, in JAMA Pediatrics by Anna Gui. And here we're looking at polygenic risk, both for autism and then for a cross-disorder polygenic risk score from the Psychiatric Disorders Consortium. And we're finding here associations in, again, it's an 8 months neural response to sort of faces versus non-faces here. This is the N290 sort of latency. We're finding that relates both to polygenic risk scores for autism, but also, and actually more strongly, to this cross-disorder polygenic score. So that may not be an autism specific finding, but there may be something about polygenic risk for psychopathology that relates to the neural response at 8 months to faces versus non-sort of faces. That's a sort of example of the sort of design we're trying to move to. Here's just some unpublished work by a postdoc, Vinnie Carter-Leno, who works with us now, very fortunately, because she's like the others, brilliant sort of too. I won't be able to tell you exactly what the analysis is here, but it is essentially an index of excitation and inhibition, and she's looking at EI imbalance, as we call it. But here she's finding that there's an interaction here between those who at 10 months have low executive attention, where if you have low executive attention, then your EI imbalance relates to your autism symptoms at the age of 3. But if you have high or intact or good executive attention, there's absolutely no relationship at all. So we're looking within that amend model at whether there are interactions between the early stage processing, such as an EI imbalance metric from an EEG experiment, with later executive attention. So these are the sorts of things that we're trying to sort of build on. I think I've said that sort of the simplistic models that many of us held to 20 years ago are wrong, and that's good, because it's important in science to have ideas and indeed hypotheses that that you either prove or sort of disprove. I think there was a bit of a struggle for 10 years where people wanted to hold on to things that were, that were because they'd been long held beliefs. We'd never had the ability to test them until we had this prospective design done at large scale. And then you want replicable results again across different labs, across different cohorts. But it's clear that we need to move to more complicated models. I mean, the satisfactory thing, as you know, for many of you in the audience, I hope, because you're developmental, experts, you're infant experts, which I'm not really. I'm a clinician. I'm the phenotype guy, so I know something about sort of clinical outcomes. I have been involved in infant work since the 1990, in the autism field only. So I'm not an infant developmentalist, but I'm very interested in developmental models and trying to understand mechanisms. And the good thing is that we need much richer models and much richer ways of investigating this to parse apart the different effects we seem to be finding. We're clearly finding, as are other groups now, there are interactive levels at which genetics, family history, neural development, cognitive development, and behavior interact. We would expect them to interact because these are not separate developmental systems. They are all, by definition, interconnected. We and some other groups are finding that sometimes you find things in the opposite direction to what we might have thought 10 or 20 years ago. And at first, again, that was a little bit disturbing, but we're less disturbed by it now because looking behavior, for example, and lots of you will know this much more than me, you have to work out what it means. And looking may tell you something about processing, but more may not be good. And, and there may be a there may be a middle ground somewhere. Some of the findings are clearly autism specific, but many are not. They're actually shared with other co-occurring conditions. And one of the things is longitudinal studies can give us suggestions of mechanisms, but we need intervention trials to test them out. So I'm coming to the close now, but I'm just very briefly going to mention the two trials that we've been involved in. Other groups have too. Other groups have too. These are a few years old now, but with colleagues Jonathan Green and our group, we've been looking at early preemptive intervention using a positive parenting sort of intervention called video interactive parenting, VIP. Looking at that at 10 months of age. So there are weekly sessions therapists have with parents in the home, and this is positive pairing, sort of, parenting, sensitizing, attenuating parents to communication in their child, helping them to be responsive, helping them to be synchronous. And this is done through video interaction and showing them the interactions and helping them learn for themselves how to promote those interactions over time. This is the intervention starts at 10 months of age, it goes on for 5 months, and this is the outcome at 14 months of age. So here we're finding at 14 months some differences, but relatively few statistically significant, but many are in the positive sort of direction, that actually at the end point of this intervention, the infants, the caregivers are less directive. There's a non-significant but marginal effect for things like the gap disengagement effect, but also the AOC. So that's the Autism Observation Scale for sort of infants. Potentially a negative effect on sort of language. And then we see the infants at 3 years of age, so some 2 years after the intervention. And here we're finding on this observational measure, the OAC and the ADOS combined into one analysis, we're finding that there are overall differences, a reduction in autism observed traits in the infants through to the age of 3 who were involved in that early intervention. Other groups are doing this, and this is a different design. It came out, I think, last month, and there's quite a lot of social media sort of reaction to it. It's led by Andrew Whitehouse, closer to you in your part of the world, from Australia, from Perth. And this is not infant siblings mostly. These are mostly infants who are identified at 12 months of age by a screen for early autism signs. But Andrew and Krishna Hudry and colleagues they used the VIP eye-basis sort of intervention sort of study, again, 4 to 5 months of weekly sort of sessions. And they found differences here on the same sort of metric in terms of reduction in autism traits, and indeed a slight reduction in those children who went on, the number of children who went on to have an autism sort of diagnosis. This is the National Autistic Society in the UK. There was a lot of criticism because it's using autism symptoms or autism as an outcome, and sort of saying that that poses ethical challenges, which I'll come back to. There's a different sort of study, but it's showing that intervention studies can be done and can help us test mechanisms as well. This I thought would be out. It's been waiting for it to be out any day for a long time, many, many weeks. It's coming out literally any day now in translational psychiatry. And this is our InterSTARS ADHD training trial with Sam Waz and many others. So this is Sam's experiments where we're using gaze contingent cognitive control sort of training to boost executive attention between the ages of 10 and sort of 14 months. Because it's a trial and it's registered, I can't tell you the outcomes, but it really is coming out within the week. We think it should have been out by now, but that's a randomized trial on infants with a family history of ADHD, looking to boost the executive attention system as opposed to the eye-basis studies wanting to boost the social engagement system. Those are ways of also testing the amend modifies model. This is a recent paper that we've been involved in with colleagues from Oxford, Alina Singh and her group, led by one of her PhD students, Ariane Manzini. And there's a lot of controversy in the autism field about different community views and about how well engaged scientists are with the community and autistic advocates, and is treatment a good thing? And it's a hot area, and one that you have to be ready to embrace and engage with the debate. This was one of our attempts within an academic journal to engage in some of the difficult sort of issues. What do we need to do ethical research in early autism, including intervention studies? And these are some of the sorts of things. Certainly, we need to make sure there's partnership and there's true, important, open discussions of difficult topics. My personal view is not everyone's going to come to the same view, and that's fine. I think diversity and the neurodiversity movement idea is important, but it also means there will be a diversity of voices and a diversity of views. And I see that as a good thing, not a bad thing. One never wants to cause offense or insult people, but one also has to accept that humans have different views about things. Okay. That's really my sort of wrap up. I hope I've given you an idea that, at least for us, the journey over the last 20 years has meant that we've had to do a bit of rethinking. We hope we're getting smarter at some of the science, but it's quite challenging. It's incredibly resource intensive, and it needs a lot of funding. It needs a lot of resource. We are embracing the complexity of autism in terms of the influences on early development, in terms of the overlap for autism as an outcome with other forms of psychopathology or co-occurring conditions. We're wanting to be more mechanistic in our thinking and not just describe what happens, but test it, either by the sorts of models we're proposing in Amend or by the intervention studies I've outlined in what we're wanting to do, both in relation to autism and in relation to ADHD. So I need to thank most of all the incredible families who stay with us for a long, long time. And some of these families have got multiple children in our studies. And one of these families I know, because the people in my group in my lab have been telling me recently, there's we call them the famous family, because I think they have 5 children in their family who are neurodivergent with a combination of ADHD and ASD. But we're still seeing them and their babies as they go up through toddlers, and we will see them, we hope, when they get to mid-childhood, and indeed we hope we'll see them through to the teenage years. Some of the infants I saw back, work with Simon Barron-Cohen and colleagues back in 1990, we saw when they were in their mid-20s in another study. So participants can be persuaded to stay on, but they are amazing. These are my colleagues, Mark, Emily, Andrew Pickles, and there's an army of people here and at Birkbeck, and a lot of funders, and along the way, a lot of fun. Thank you for inviting me, and it's been a pleasure to talk to you.